So thanks very much for the invitation. There was no traffic on the way here, so I got here radically early because I was expecting traffic, but I guess it's a holiday. So yeah, today I'd like to kind of break up the talk into two sections. The first is more thinking about some of the things I worked on in the past, but we're still thematically interested in terms of the mechanisms of learning and how subcellular processes are involved with the formation of memories through long-term plasticity. And then the second part will be, thematically completely distinct, but I think we're still thinking about some of the same type of mechanisms in terms of circuit inhibition. So how do we think about the neurological basis for learning? And this is kind of how I define it, is that when you're learning something, there's a dedicated circuit in a particular part of the brain that's activated and responsible for driving that type of learned behavior. And that learning occurs following, very simply, a change in either the synaptic or signaling properties of neurons within that circuit, best known as plasticity. So how might it actually work in real life? Let's see if I can get rid of this thing. Maybe I'll take the X out of this. Sorry. Hopefully it's working now. There we go. So here is an example of a particular type of learning considered motor learning. So it's a couple of years ago, it was about 10 years ago, this subway station in New York City had an anomaly with its entranceway that really allowed for some kind of real-time observation of what a motor learning example might be in humans. You'll notice that some people have a lot of trouble with this step, which is about a fraction of an inch difference from the other steps. And some people have a really hard time with it. So in my mind, I'm thinking that some of these people are tourists, are getting off this step for the first time. Some of these people are maybe locals, they're used to the step being off. I think there's one really good example near the end of that. This guy really almost bites it. Here we go. His friend looks at him like, what, you didn't expect that? So how could we study this process? Well, there's many different ways that as neuroscience we can think about this. We could think about it in big cortical loops or in big circuit-centric sort of models. But our lab, as a theme, thinks about these kinds of things or wants to at least study these types of things in sort of a neuron-centric manner. So as a lab, we kind of want to understand what is it about different subsets of neurons, what is this sort of set of signaling functions that they need to perform, and how do they do that to perform these sort of processes. And even more specifically, compartmentalization of signaling within neurons themselves and different types of neurons is sort of a key feature that we look at in terms of learning, memory, and also in disease models. So a really good cartoon example of how compartmentalization of signaling works within neurons is a single action potential can fire near the soma, actually in the proximal axon, and that red compartment is actually the axon. You can see the signal is much sharper, and the signal is much wider in the somatic compartment. Let me see if I can get this laser pointer. And then in the dendritic compartment, this is the same action potential, but it's broadened out. So why is it that these signals are sort of expressed differently throughout the neuron? What purposes do they serve? Would that serve? And our approach is, just to give you a little bit of background of some of the techniques we're working with. Just about everything I'll show you today is from acute slices from adult mammalian circuits, so mice, either cerebellum or cortical slices. We use a number of physiological approaches to look at cellular function, whole cell patch clinical electrophysiology, and another number of different electrophysiological approaches to photon microscopy to look at subcellular and cellular processes. A little bit more in depth, we use voltage sensitive imaging, so sometimes dyes, and more recently we're now using genetically encoded voltage indicators, fluorescence guided patch clamp, which helps us to patch onto very, very small regions of neurons. And I think I even have an example of ion channel photolysis, so we can actually block different conductances in a focal manner within the neurons to kind of dissect out where they exist and what their purpose is. So as I just alluded to, the first part of the talk is going to be solely focused into the cerebellar region, so the cerebellum. And the reason we use cerebellum is because it's a fantastic model for plasticity and learning. It's sort of in terms of mammalian circuits anyway, it's a simplified circuit. There's a lot of advantages to looking at cerebellum. Its major function has really been well defined, and there's a number of motor learning pathways that have been identified, such as eye blink conditioning and vestibular ocular reflex, or VOR. There's very well-defined circuits and cell types in the cerebellum. The Purkinje cell, the PC, is the major output and really the only output of the circuit. And these parallel fibers, which are emanating from the granule cell, those are the most numerous neuron actually in the mammalian brain. And basically what they are conveying is context, sensory motor context. So if you're moving your arm, there's going to be a certain number of parallel fibers that are sort of firing during that process. And the climbing fiber, and so that is what's thought of as the cell types that's really responsible for conveying error signals. So during a movement, if you're activating all these parallel fibers and something, a sensory motor mismatch happens, for example, you're on that step and you almost trip, then it's thought that those climbing fibers are going to fire, and they are extraordinarily strong synapse. So that's the parallel fibers in green. That's what a typical sort of synaptic input into a Purkinje cell would look like from the parallel fiber. And the climbing fibers in red. If you're recording from the Purkinje cell, that PC, then you'll get a very strong complex spike occurring. And how does plasticity classically thought to occur within this circuit? So that happens when you have a near coincident pairing of these two pathways. So you're activating some of those parallel fibers, perhaps you're in some sort of a motion sequence, and you make that error, and then you get an error in your environment. And when you have the parallel fiber input and the climbing fiber input in this close coincident in time, then that's thought to trigger plasticity in the circuit. Interestingly, the climbing fibers are carrying this plasticity, or they convey plasticity in what I think of as a very compartmentalized manner. So very dendritic-centric mechanism. It's completely subcellular, excitable phenomenon, distinct from the soma. And these are just some sort of paraphrasing from really classic papers in the field. And I can show you that through a single experiment, how that looks. So if we can actually electrically stimulate all these pathways, record from the Purkinje cell, and at the same time also record from the dendrite. So these are whole cell patch clamp recordings that are happening simultaneously at the two different regions of the same neuron, while either you can electrically stimulate a climbing fiber or a parallel fiber. So this is the really striking compartmentalization of these signals here. So you see the Purkinje cell at the soma. If you fire the climbing fiber just one time, you see these rapid sodium spikes that happen. If you look at the recording at the dendrite, the Purkinje cell dendrite has this completely different signal. It's almost like completely separated temporally as well, if you overlay the two. And actually what's happening at the dendrite are these are actually calcium spikes, as opposed to the soma, which are being conveyed by sodium spikes from the axon. So if we look at a recording now from the Purkinje cell and look at what happens classically with parallel fiber stimulation in close opposition in time to a climbing fiber. There's classic papers now that show that this results in LTD, so classic cerebellar LTD. What we know now is that there's an enhancement of calcium entry into the dendrites of these Purkinje cells, specifically in the spines of these dendrites, that allows for that plasticity to occur. So what does it look like? We can actually do a calcium recording from single spines. So this is using just a calcium dye, like a flow 5F. And with looking at that single spine during a climbing fiber event, you get some calcium entry. And if you look at that spine during parallel fiber plus climbing fiber, you get some calcium entry during the parallel fiber and some during the climbing fiber. Now, if you look at what would be expected from the sum of the parallel fiber and the climbing fiber by themselves, that's what this looks like here. That's sort of the arithmetic sum of the predicted. But when in reality, when you have this parallel fiber and climbing fiber signals coming in nearly simultaneously, you have this superlinear increase in calcium that happens in the spine, and that's what's thought to underlie the expression of LTD. So in reality, there's a major issue with how this could work. And the reason that is because climbing fibers are firing spontaneously at about 1.5 hertz. So the question becomes, why is it that learning mechanisms are not saturated then? Because you're always getting these complex spikes in the dendrites. So how are errors and thus motor learning reliably encoded in this case? So one solution is this active regulation of dendrites, right? Now in an artificial sense, we can patch a dendrite, and you see we stimulate a climbing fiber, like I showed you before, and you see this big calcium spike event, actually 3 calcium spikes that happen with a single climbing fiber firing. Now if you just hyperpolarize that dendrite with your patch electrode, if you've got good whole cell access, you actually can reduce the amount of spikes that occur just by hyperpolarizing that dendrite. The only problem is that this is very difficult to achieve because our brains don't have patch electrodes attached to every Purkinje cell, right? So we need a circuit mechanism to modulate these particular signals if we want to be able to modulate how much learning is occurring in those dendrites. So it just so happens that there's a potentially perfect circuit mechanism in the cerebellum in which we can modulate these signals through inhibition onto the dendrites. So one cell type I didn't mention is this molecular layer interneuron that exists in the cerebellum. So those molecular layer interneurons are inhibitory GABAergic cells, and they make numerous synapses onto the dendritic tree of these Purkinje cells. Otherwise, it's known as MLIs. There's a number of them that make basket synapses, and a number of them that make synapses, again, along the entire dendritic arbor. And so there's these kind of circuit analogs in cortex and cerebellum that you'll see that inhibition occurs along the somatic axis and the dendritic axis as well. So these kind of things exist in cerebellum also. We were really fortunate to form this collaboration at the Max Planck with the Haruki Taniguchi's lab, they're looking for drivers of interneurons in cortex. And they had one mouse called the c-Kit-Cree mouse that basically wasn't any good for them, but they were looking very, very quickly at cerebellum and said it looked like there's something interesting happening there. And so we took a look at what cell types were actually able to be, what gene expression we could drive with this mouse in the cerebellum. And it turns out it was a completely pure population of interneurons, which is really fortunate because we want to be able to do all this but also drive the activity of interneurons using optogenetics or chemogenetics. And so not only does the confocal image look good, but we also went in and did all the electrophysiology in this mouse line. In molecular layer interneurons, when you turn light on, after expressing virally channelrhodopsin in the c-Kit-Cree line, you see that you get activation and you get firing. But in every other cell type, in the cerebellum, if you patch Purkinje cells, granule cells that have those parallel fibers attached to them, those will never fire. So it's a very nice, pure model that we can use to drive interneuron firing during this whole process to kind of model the system. So what is the effect of inhibition on these climbing fiber signals? And we can ask that by looking again at our dual recording. So we have a recording at the soma and the Purkinje cell, and we'll have also a recording going, whole cell recording going at the dendrite. And then we can again stimulate the climbing fiber to elicit that error signal that we think is occurring during motor errors. And again, when you stimulate that climbing fiber signal, you get that complex spike that occurs at the soma and the dendrite. Now, the only thing we're going to do on top of this is do coincident pairing of that interneuron spiking during this whole experiment to see what happens. What was interesting to us is that if you did coincident pairing of these interneurons and look at the somatic signal, there's actually not large differences. If you really crank up the firing in the interneurons, you might be able to knock out one of the sodium spikelets, but often you'll see no difference like from control to light on. So this would be with interneurons firing. Now, when we look at the same exact experiment in the same cell, but now we're just looking at the dendrite recordings, what we see is that those multiple calcium spikes in the dendrite are largely reduced. You can go from 3 calcium spikes down to 1 calcium spike just with having coincident interneuron input into the dendrites. So this is kind of the potential way in which the cerebellum might be able to regulate against aberrant learning, essentially, or excessive plasticity, in other words. We can also look at this experiment completely optically, or nearly completely optically, by just switching some of the parameters around. So we, instead of using electrical recordings now, we can look at it using calcium imaging in the spines. And we just switched, and this is the nice thing about having a good Cree driver line mouse, because we can now use a red activating channel rhodopsin. And the reason we use a red, just a little trick to the trade, the reason we like to use a red channel rhodopsin here is because if you're doing 2-photon microscopy, your PMTs are super sensitive, so you would have to shutter out that red optogenetic light normally, or that optogenetic blue light. But in this case, since we're using this red light, we didn't have to shutter at all, and thus we're not losing any of the calcium signal that we really want to get out of this experiment. And so you can see kind of an analogous experiment to what we just did electrically. Now we're just looking at it in terms of the calcium imaging at the spines to prove essentially, okay, are we really affecting calcium entry during this process? And so this is just the control traces again. So you see the complex spike that happens at the soma, and there's that dendritic calcium spike that happens out in those distal dendritic spines. And so this is just after a single climbing fiber input into the dendrite. If we do the same experiment, and now we're activating our interneurons optogenetically in a coincident manner, and we see again, this is just another example that with and without inhibition, those somatic calcium signals are not changing. However, those dendritic calcium, the somatic electrical signal is not changing. However, that dendritic calcium signal that we think is so important for the expression of plasticity is largely reduced. And so, of course, what we know about neurons and interneurons specifically is that they fire at different rates depending on their input and how strongly their inputs are. So we ask the question then, if we change the firing rate of those GABAergic interneurons during this process, can we thus modulate the calcium entry in basically a stepwise manner? And that's one of the beauties of optogenetics, is that you can actually do that So we did a number of control experiments to show that we can get reliable firing at different rates with different light power, essentially. So we were able to make the interneurons fire at what we call light or moderate rates or high rates, like 30, 60, 100 hertz, for example. And what you see that is that if you change the rate at which interneurons are firing coincident with those climbing fiber events, for example, this is the control trace, and then this would be with 30 hertz inhibition, and this would be with 100 hertz inhibition. Then you actually have a way to essentially titrate the calcium entry during the same electrical input through the use of circuit inhibition. And if what that immediately starts you thinking about is, if you look at cerebellum or cortex, it doesn't really matter. It's all about magnitudes of calcium in terms of what type of plasticity you get or what magnitude of plasticity you get. So we're able to modulate the magnitude of calcium. And in addition to that, although I'm not talking about it in this context, people often think about the magnitude of calcium entry happening perhaps through an NMDA receptor as a trigger for excitotoxicity. So this is another way to think about why this might be important. But this is just a kind of a schematic from Kozmans et al. Showing that in cerebellum, when you have very high calcium entry, or low, sorry, just slight calcium entry, then you tend to get LTP. But if you get very strong, that supralinear calcium entry that I showed you before, you get LTD as a result. And so we just basically took this entire environment and switched it to a plasticity experiment and said, okay, depending on the strength of inhibition, are we going to be able to modulate long-term plasticity in that way? And so this is a classic experiment again, where now we're just looking at the plasticity instead of calcium. So this is classic cerebellar LTD. Remember, if you're pairing parallel fiber inputs with a single climbing fiber input, then that results in LTD in the cerebellum. And this is just sort of the time plot showing that this is during the conjunctive stimulation of parallel fiber and climbing fiber, you get this long-term effect of a depression between that parallel fiber to Purkinje cell synapse. Now the only thing we're going to do is add inhibition during that climbing fiber spike, just like I showed you with all those calcium experiments. And what I'm showing you first is this inhibition happening at what we are considering moderate firing, so 60 hertz firing, just during that climbing fiber pairing. So we have the same exact experiment. The only thing that's different is this inhibition that's occurring coincident with the climbing fiber. Now, instead of LTD during this experiment, we actually saw no plasticity on average in the long term. This is the time plot. You see that before and after, you're having essentially no difference. So that harkens back to the initial problem we were thinking about, which is how is it that if you're getting calcium spikes in dendrites all the time, which is true throughout the brain, how is it possible that we can not always undergo plasticity and overwhelm that cellular machinery that's involved with undergoing different forms of long-term plasticity? And this is sort of demonstration that with moderate inhibition, which is happening all the time in circuits, feed-forward inhibition, et cetera, that you can basically eliminate that process from happening. It's also kind of an important thing to always think about. It. During many LTP protocols, people tend to block inhibition all the time. So if you leave inhibition intact in the circuit, that's going to result in something different. Now, a couple slides back, I told you that actually moderate calcium entry actually results in LTP. So the next thing we tried was very strong inhibition, which should essentially block as much calcium entry as we possibly could during that climbing fiber event. And now it's interesting, you see that you went from no plasticity to actually you changed the sign of plasticity to LTP, and that's shown in this graph here. Actually, the gray circles are if you only use parallel fibers. So if you just have parallel fiber, high frequency input, that actually results in LTP. And if you basically use strong inhibition here during that climbing fiber input, what we've basically done is say to the cell, ignore that climbing fiber input, because the result is exactly the same. So in total now, what we've demonstrated is that you can have kind of low, medium, or high calcium entry during the same signal based on the circuit inhibition. And the consequence of that is that you can basically titrate the sinoplasticity and the amount of plasticity you get. So I basically just included this slide here to say if you're really interested in this, you want to see the in vivo experiments we did about it. We took this whole slice environment and we brought it to the in vivo level using fibers and optogenetics and a cerebellar specific behavior called vestibular ocular reflex. But basically, in the interest of time and everyone's attention, I think that was enough cerebellum for one talk. But the take-home message here is essentially that you can do the same thing in the living animal and you can modulate the type of learning that animal will express, either kind of in one direction or the other, based on the activity of these interneurons in the circuit. That's just a video of VOR. So it happens completely in the dark in this case. And we're using infrared light to look at this. And so it's just a completely beautiful cerebellar specific behavior that the mouse basically has no choice but to undergo because it's basically just completely motor. So the first take home here is that inhibition in the circuit, depending on how much you have, is going to allow for graded expression of plasticity and learning. And we think that this might occur in different circuits in different contexts as well. One good example beyond that may be that, for example, place cells in hippocampus, you see that when they're undergoing some sort of learning, you'll see a plateau potential that happens in that dendrite. And our hypothesis would be that circuit inhibition during that event would change the expression of essentially place fields. So it's just kind of a different context, but the cellular mechanisms are sort of lining up. So for the second part of the talk, I wanted to introduce a new topic we're looking at in the lab, and we're kind of looking at a very similar cell type. We're looking at a fast spiking interneuron. In this case, we're moving to cortex. And we're looking at the physiology of those neurons in the context of Alzheimer's disease. Let me give you a little bit of background about why we might be interested in doing that. So the prevailing theory, now switching gears to a completely different topic, very rapidly, a little bit of background. Sort of the prevailing theory underlying the etaology of Alzheimer's is that over time, these toxic accumulations of protein, these amyloid plaques, and then tau aggregations following, that sort of build up over time, and they result in neuronal death and synapse death, and eventually, of course, the sort of connected cognitive decline that happens. However, I think recently, as the field starts to shift and the more and more evidence coming out, is suggesting that those protein aggregates are not necessarily the causal determinants, especially of the initiation of the disease, but rather sort of an end effect almost. And this is most perhaps strongly demonstrated through the failure of some of these recent drug trials that work quite well to reduce soluble amyloid, but they don't really stop cognitive decline, except I know there's some evidence that that's may be somewhat effective coming out recently. So where our interests kind of line up to this is that we really want to understand, okay, what's happening really early on in the disease process. So what's happening in the so-called pre-symptomatic, pre-plaque stages of the disease. And there's actually quite a bit of literature out there now about what's happening in circuits, hippocampus and cortical circuits in models of AD, and it's also humans with mild cognitive problems leading to Alzheimer's. And so one of the hallmark features appears to be that there's an imbalance of excitation and inhibition that starts to occur in the circuit, and such that you get this over-excitation happening in the circuit. And so I said not just a hunch, there's lots more than here. There's tons of literature. And so interestingly, it appears that since we're really interested in different neuron types and how they work and how what are their conductances and things like that, there's a lot of literature that's really striking to us, showing that in different models of AD, there's one particular type of interneuron, this inhibitory interneuron again, that appears to have a physiological deficit early on, and kind of even can spare the other types of interneurons, like somatostatin interneurons, for example. And so that interneuron is this fast spiking, or PV, if you guys are more familiar, this PV interneuron depicted in blue here. And so interneurons in cortex are often sort of characterized on their genetics, but also their axonal targets. These guys tend to target the the proximal dendrite and somatic compartment of the principal neurons in the circuit. And this is now kind of classic work from the PALUP group showing that in a model of AD, this is a mouse model of AD, that you can get differences in action potential firing in these cortical fast spiking interneurons. And all the other groups have now shown in different brain regions very, very similar phenotypes, although there's some disagreement about exactly what happens. There seems to be an agreement that the susceptibility to physiological deficits are really strong in these fast spiking interneurons. So of course that kind of fits into our wheelhouse perfectly, because we're really interested in what are the cellular and subcellular mechanisms that fit into, or that are responsible for action potential firing, this fast action potential firing in these cell types. And so, no doubt, the most important thing that's determining, the most important factor that's determining this fast spiking phenotype in these cell types are their conductances. They're endowed with these really specialized fast ion channels, like NAV1 and KV3, for example. We kind of have a historical interest in KV3 for a lot of reasons. So we are now sort of looking into these channels, along with the NAV channels as well, and sort of seeing how might this fit into the context of AD in both sort of a therapeutic manner, and also some of the mechanisms, the deficits that might be occurring in these cell types also. So this is really, these conductances that drive spiking in these neurons are well, at least the general families are well known for a long time. So KV3 channels are these sort of fast delayed rectifier channels that are known to play a strong role in the spiking activity of these cells. You can see this is from 1999. Basically, the experiment just dumps into the bath of this slice recording a low concentration of TEA, which is quite selective for KV3, not completely, but it's pretty good. And so these kind of things have been known for a while, like there are certain types of conductances that are overexpressed or specially expressed in these cell types that allow for that phenotype. And so like what's important in the context, I should mention again, like why is this important in the context of AD, is that if you're losing firing in these neurons, or other diseases as well, if you lose firing frequency, if you lose amplitude of action potentials, then essentially that will disturb the inhibition in the circuit. It has the potential to lessen the inhibition that those postsynaptic pyramidal neurons are receiving. And so that could lead to an overexcitable circuit in general, which is what is seen. So this is just the results from our lab showing, 20 years later you can get the same effect, which is always good. And so this is kind of dumping a low concentration of KV3 blocker into the bath. You get a broadening of action potential width, you get slowing of spiking. So we know certain conductances are responsible for the firing of these cells. But also, there are other factors that sort of fit into this picture that you can't really garner from these kind of global pharmacological experiments. And these are things that we've been working on for the past few years, is that conductances, really important conductances, are hard to study in these cells and all neurons because they're localized in these subcellular compartments of neurons, right? So not only is our particular channel types like Kv3 important, but also their localization within the axon is important. I kind of jumped the gun. So the axon initial segment in this case right here is where action potentials are actually starting. That's where they're initiating. And this is true for basically all neuron types, but not until recently did we kind of confirm that this was also true in these fast spiking interneurons. And the way that we were able to determine that is true is through the use of just a regular sort of patch clamp experiment and simultaneous voltage imaging down into the axon. So that allows us to see the signal happening in nearly simultaneously in both locations. And what you see is that the axon initial segment, this signal in red happens before the somatic signal. And if you look at distance, and this bar is actually ankyrin G staining, which is basically a cytoskeletal protein showing you where the aggregates of many channels and other proteins happen within the axon. You can see that the somatic, sort of AP latency is lowest at around 10, maybe 15 microns, 12 microns away from the soma. So basically we're hypothesizing that the channels most important thus for action potential initiation and maybe even high frequency firing are kind of situated at this area. And we can actually test this, which is really fun, because we're able to focally block ion channels in different regions of neurons. So we can use this caged compound that can block Kv3 channels called ruthenium 4AP. So ruthenium is actually the cage. And the nice thing about this ruthenium cage is that 2-photon light, which has a very small focal volume, can actually uncage this Kv channel blocker. And so if we just then say, let's situate our uncaging light around this 10 micron stretch of the axon, will we actually be able to, or will we actually affect firing of that axon? And the answer is pretty amazingly yes. If you block just the channels that are situated along that axon initial segment region, then you see this reduction in action potential firing in these fast spiking interneurons. So it's also be very important to look at sort of the conductances throughout the somatodendritic compartment as well, and other groups have started to do this as well. One thing we're really lacking on, beyond just knowing what families of channels are in these neurons, are actually the true molecular identity of these guys. And the reason is that these are just really low throughput experiments. And so it requires that you kind of go through knockouts or some other method which you can get molecular identity and then find it in a localization dependent manner as well. But we've done some of these experiments too by combining knockout animals with very precise subcellular recordings of these ion channels. So you can see this is our axon pipette. The open tip diameter is about 200 to 300 nanometers. So we can actually go and use fluorescence guided under the 2-photon and then take recordings from these interneuron axons at the, for instance, presynaptic layer at the axon initial segment. And so the take home here is that we were able to identify not only the families of channels, but actually some of the subunits that predominate in the axon. So knowing the molecular identity of these things is going to be really important if we want to use gene therapeutic methods in the future, for example. So we see that there's 4 subunits of Kv3. We went through a few of these things, but it looks like Kv3.4, this one of 4, is dominating in these fast spiking interneuron axons. And it just so happens that through biophysical modeling, it's showing that that particular channel seems to be really good at fast spiking as well, so it wasn't a big surprise. It looks like it might actually heteromerize as well, but once you lose Kv3.4, that seems to, for whatever reason, we don't actually know why, knock out all those Kv channel conductances presynaptically or in the axon. So then we ask the question, okay, we know the molecular identity of the Kv channel, Kv3 channel that we think should be involved with high frequency firing, the naturalistic firing of these cell types, but we actually haven't done that experiment molecularly. So what we did next is design an AAV CRISPR. So this has got an SA-Cas9. It's like a next generation Cas9 that you can fit into an AAV vector with the sgRNA against Kv3.4. And then we just injected that into layer 5 cortex and waited a couple weeks and then performed recordings from these interneurons. Because they're TD tomato positive, we can actually identify them for recordings that way. And initial data is really exciting, I think, because it looks like just from the loss of this particular subunit, you're getting this dramatic difference in firing frequency in these neurons. A lot of the times you have different subunits for a particular protein that are very similar, and one can kind of substitute in for another, but it looks like in this case, this particular molecular subunit is really important for that phenotype. So going all the way back to AD and neurodegeneration, we thought that it would be a good proof of principle to show that if we perturbed interneuron activity through knocking out this gene, would that have an effect on essentially synapse fidelity of the excitatory neurons in the circuit? So the question we ask is essentially, if we lose this subunit in interneurons, does that have any effect on synapses, which is essentially, I think, the most agreed upon early indication of a problem in the circuit in AD and other diseases is synapse loss. So the way that we did that experiment is just to essentially combine our control experiment or our CRISPR experiment against Kv3.4 with a marker for the postsynaptic pyramidal neurons. This has a promoter, CAMK2, that's specific for those cells, and then it just expresses YFP, so we can visualize the spines on those dendrites after the experiment. And what we found is that compared to control injections, when we use our Kv3.4 CRISPR, viral CRISPR method, just after 2 weeks, you get a reduction in postsynaptic synapses and spines. And it's a bit stronger in layer 5 as compared to layer 1. And also you get some difference in spine length in layer 5, but not layer 1. The differences in spine length we don't know exactly why that's happening, but other groups have shown that synaptic activity changes, like through NMDA receptors, for example, can start to change the shape of those spines. And the reasoning, the possible reason why you're getting differences in layers is that we're injecting in layer 5, the PV interneurons in that layer are contacting the somatodendritic region, so you might expect a more proximal effect. But it's interesting that you're still getting a very distal effect in those distal dendrites up in layer 1. That could be through regulation of spiking of the cell in general, or maybe other reasons. So moving forward, I guess our hypothesis will be, or one of our questions will be, of course, let's look at this, let's look at that conductance naturally in models of AD, so that's what we're doing actively now. It's been shown that overexpression of different sodium channel types can improve some of the function in AD models, except working with sodium channels is very difficult because essentially you have to do a cellular therapy in order to get that to work because they're such huge molecules. The advantage of using Kv3 in this context may be that you can, of course, overexpress these channels using AAVs, as shown here. So we're actually able to overexpress Kv3.4 itself. In fast spiking interneurons. And you see this kind of really interesting cellular effect that I didn't really have time to go into today, but it does tend to sort of aggregate to different parts of the axon, which could have interesting sort of consequences for inhibition. So general summary is that these particular subunits in fast spiking interneurons appear to be required for the spiking phenotype in total of the cell, and that acute knockout of that sort of very specialized conductance leads to synapse loss fairly rapidly in the adult cortical circuit. So we kind of want to bring it, attempt to look at this in the reverse order, and that's what we're doing now is if we overexpress this channel, can we halt some of the phenotypes we're seeing in the model of AD itself. Thanks for your attention. Happy to take any questions.
You mentioned you're looking now at the AD model. Can you tell us if you're seeing anything and what model you're using?
We will know soon, I guess, but actually what we're attempting to do first is recreate the J20 model, but virally in an acute way. So that would be the Indiana-Swedish mutations, but instead of having the mouse grow up with it, we're gonna overexpress that HAPP and see how that works. But I think the alternative to that would be, and the reason we started there is because, I mean, the PALIP stuff, they started using that in that model, and they see the interneuron deficits in that model. So we're just trying to not reinvent the wheel too much. But I think we're weeks away from getting our first information there.
So it seems that for the inhibitory neurons to actually regulate plasticity in the cerebellum, there has to be this fine temporal precision between the activity and the neurons that target the dendrites, I believe. Do you see evidence of that?
Well, actually, we kind of overdid it with the temporal precision because we were able to do that. But you're right. Actually, normally it's not going to be that beautiful in terms of its feed forward. Actually, what actually is happening in vivo is that the parallel fibers are activating molecular layer interneurons, which is leading to feed forward inhibition all the time. So, but we did do experiments in vivo which were kind of naturalistic, and then we just turned on or we turned things off in terms of turning off the interneurons, and then you can still perturb learning in that way. The reason we were kind of doing it the way we were doing it is just to be extreme about it, essentially. So the interest also goes then to, I mean, it always goes to the next layer of complexity, but there's plasticity between the parallel fiber and the interneurons there, and that may play a role in that whole global system as well. Yeah.
I've read a paper in Cerebellum where the timing precision between parallel and climbing fibers resulted either in long-term potentiation or depression, depending on the timing.
Mm-hmm.
But it doesn't really go into like the mechanism to show that it's there. So like, do you think that the MLI might play a role in that, or
yeah.
is that the mechanism maybe?
So I think most of the studies that look at that tend to be blocking inhibition, but ultimately I think where things occur, for example, in time, may have a lot to do with sort of the electrical nature of the dendrite, and there's some groups that would say, okay, there's also a chemical like mGlu-R, but to my mind, we've actually looked at some of these experiments where basically, how do you get LTD, and what's the timing got to do everything, for example? Well, the parallel fibers are depolarizing the dendrite, and such that it's almost priming the dendrite before the climbing fiber comes into. When that climbing fiber hits, you're getting that sort of massive triple calcium spike that happens. And I've actually shown this in a couple of ways that that could be one of the major mechanisms there. That would be why, if you get a climbing fiber first, you don't have that previous depolarization that's happening that's sort of priming that dendrite. And I guess the other side of the card is that some people think that mGluR6 is required, like the Wang group a long time ago now showed that they block that, and then they can't get LTD. But I think it could be multiple mechanisms there. But ultimately, the thing that's causing LTD in cerebellum to happen is a larger magnitude of calcium, which is interestingly the opposite of cortex, which is this, and it kind of makes sense in the way that the cerebellum and the cortex speak to each other. But if you get this massive entry in cortex, you get LTP. So ultimately, regulation some way, whether through its internal stores or dendritic active properties, that magnitude of calcium seems to be the big, what's determining things.
So in your CRISPR-Cas experiments, have you patched the pyramidal cells to see if they're more excitable?
Yeah, we actually have, and interestingly, so they're getting an increase in spontaneous EPSCs happening. So what the mechanisms that you're getting to that point, we don't know yet, but you are getting that happening.
Like
Yeah, exactly. And we don't know, in terms of the CRISPR-Cas9, we're not sure what's happening with the spiking yet, although we've done the opposite where we've had overexpression of the Kv3.4 and looked at the pyramidal neurons, and they're actually spiking a bit less. Now, whether that's like cell intrinsic or homeostatic, like we don't know where that's coming from yet, but it looks like things are kind of, if you upregulate those channel subunits, then overall the circuit seems to want to get quieter, I guess.
kind of follow-up to that.
Yeah.
When you do the overexpression, it looked like in that image you showed that they were out on the boutons.
Yeah.
They weren't localized to the node, right? Which is
Yeah.
one of your key things that you were setting up, right? So
No,
how
that's
are you
great.
gonna get a localization signal on those together?
No, you took the cat out of the bag there, because like we did a number of these experiments in cerebellar interneurons, and this is actually a cerebellar interneuron. And so we have to actually look at axon initial we have not looked at that experiment in the cortical fast spiking interneurons yet, but the hypothesis is that in those cortical fast spiking interneurons, those Kv3.4 channels are going to be chock full within the soma and the axon initial segment. But we still have to do that. We have done some outside-out patches from the interneuron somas, and there seems to be some loss of conductances at the soma, which is kind of interesting. Like this is just another example of how within brain regions, neurons that seem to do the same thing and behave similarly, they just still have different molecular and spatial organizations. So they just have to find a dynamic range, which they're happy in with what that circuit's trying to do and stuff. So it's kind of crazy.
Follow-up question. So when you do the technical question, when you do the experiment, do you, like on the cell model, do you need to grow the cell in a particular, like your favorite or like the commonly accepted medium, and in a certain like 3D, like version of
The best thing about this is that I don't have to grow anything because it's all ex vivo. Everything's from slices. Some people might think that's bad. They don't want to do that, but I'm just happy not to have to change a medium every day and stuff. Yeah, it's all ex vivo though.
the cells from like mice.
It's all mice, yeah. Mm-hmm.
Okay, let's
Thank you.